Welcome to this explainer. Today, we're taking a truly fascinating journey deep into the biological architecture of the stress response. I mean, we all know what stress feels like, right? The racing heart, that tight chest, the sudden hyperfocus. But what's actually happening under the hood? Your brain is this absolute master coordinator, constantly scanning the environment and reacting to threatening stimuli. And honestly, it doesn't even matter whether there's a literal predator in the room or just an imagined worst-case scenario playing on loop in your mind. Today, we're going to teach this in depth. We're going to map out the precise neural and hormonal systems that spike your vigilance, mobilize your energy, and literally keep you alive. Okay, let's dive into this. Every single one of us has experienced a moment of sudden panic or deep anxiety. Maybe it's a missed deadline at work, a sudden loud noise, or, an unexpected medical bill in the mail. But instead of just looking at the outward psychological experience, I really want you to look inward. What exactly happens inside your brain when you panic? Like, how does a single stressful thought trigger this massive cascade of chemicals that physically alters the state of your entire body? Well, to understand how to manage stress, we first have to understand the deep cellular biology that creates it in the first place. So here's our chronological roadmap for today. 1. The chain of command. 2. The gas and the brakes. 3. Fight or fright. 4. When stress becomes toxic. And finally, 5. Calming the system. Section 1. The chain of command. The humoral component. This first part is all about how we fundamentally react to pressure. Before we get too deep into the weeds here, we need to define the absolute foundation of your biological stress pathway, and that is the HPA axis. HPA stands for hypothalamic pituitary adrenal. You can think of this axis as the body's primary humoral chain of command. And when we say humoral, we just mean it involves the secretion of chemical messengers, hormones, into the fluid systems of the body, specifically your bloodstream. The HPA axis starts deep inside your brain and ends with this massive flood of hormones entering your general circulation, basically broadcasting a state of emergency to every single cell in your body. Since we really want to teach this in depth, let's walk through the exact 3 steps of this biological chain reaction. Step 1 begins in the hypothalamus. Under stressful conditions, these highly specialized cells, called parvocellular neurosecretory neurons, which live in a specific region called the paraventricular nucleus, they just spring into action. They release a peptide called corticotropin-releasing hormone, or CRH for short. This CRH travels a very short distance into the portal circulation of the pituitary gland. Moving right to step 2, within mere seconds of receiving that CRH signal, the anterior pituitary gland secretes its very own messenger, adrenocorticotropic hormone, or ACTH, directly into the bloodstream. And finally, step 3. That ACTH travels all the way down to the adrenal cortex. That's that outer layer of your adrenal glands, sitting right on top of your kidneys. The adrenal cortex receives the ACTH and triggers the release of cortisol, which is a super powerful steroid hormone. Cortisol then circulates all the way back throughout your entire body and brain, binding to receptors that actually influence your gene expression and fundamentally alter your cellular metabolism so you can deal with a threat. Pretty wild, right? Section 2. The gas and the brakes. Regulating stress. Now, the HPA axis doesn't just operate in a vacuum. It needs strict management to keep things running smoothly. And this brilliantly illustrates how it's regulated in a push-pull fashion by 2 distinct brain structures. Think of them as the gas pedal and the brakes. The amygdala is the stimulator. That's your gas pedal. It processes fear. When you perceive a threat, sensory info enters the basolateral amygdala, relays to the central nucleus, and then sends a signal through a pathway called the bed nucleus of the stria terminalis. This pathway directly activates those CRH-releasing neurons in the hypothalamus we just talked about. Boom, the stress response is initiated. But obviously, you can't stay in a state of panic forever. That is exactly where the hippocampus comes in as the inhibitor, the brakes. The hippocampus contains a very high density of glucocorticoid receptors. These are basically highly sensitive detectors that monitor the amount of circulating cortisol in your blood. When cortisol levels get high enough, the hippocampus detects it, activates, and sends a negative feedback signal right back to the hypothalamus to shut off the CRH. It's essentially telling the brain, okay, we have enough stress hormones, the threat is handled, turn off the tap. Section 3. Fight or flight. Autonomic nervous system. Now, what's really interesting about this is that simultaneous with the HPA axis activation, your autonomic nervous system kicks in for immediate physical survival. It operates essentially like a seesaw, working on this concept of reciprocal inhibition. On one side, you've got the sympathetic division. This is your action center. It instantly increases your heart rate, boosts your blood pressure, dilates your pupils so you can see better, and mobilizes glucose reserves for immediate muscle energy. On the exact opposite side of that seesaw is the parasympathetic division, which normally handles calm, non-essential functions like digestion, resting, and physical growth. Because of reciprocal inhibition, when the sympathetic system spikes to fight off a tiger, the parasympathetic system is entirely depressed. I mean, you simply do not need to be digesting your lunch when you're literally running for your life. Section 4. When stress becomes toxic. Mechanisms of damage. So, this evolutionary setup is absolutely brilliant for running from literal tigers. In acute, short-term scenarios, cortisol is an incredibly helpful substance. It aids survival. Biologically, it works by increasing the entry of calcium ions through voltage-gated channels in your cells. This influx of calcium dramatically enhances metabolic activity, ensuring that your brain is hyper-alert and deeply focused on the immediate challenge in front of you. It is just the perfect short-term survival mechanism. So the crucial point here is, what happens when that short-term mechanism never actually turns off? If stress is sustained and unavoidable, high levels of cortisol literally become toxic to the brain. Research in both primates and rodents has mapped out this frankly terrifying vicious cycle. Chronic exposure to cortisol causes the dendrites, which are the branching communication lines of your neurons, to just begin to wither. Eventually, this excitotoxicity leads to the literal death of neurons, particularly in the hippocampus. And why does that matter? Well, remember how we discussed the hippocampus acting as your brain's brake pedal? If chronic stress kills off your hippocampal neurons, your brain loses its ability to send negative feedback to the hypothalamus. Without those brakes, the stress response just escalates unchecked, pumping out more cortisol, which in turn kills more of the hippocampus. It's a literal biological downward spiral. But actually, there's a fascinating flip side to how our brains adapt, and it's heavily influenced by our earliest life experiences. In studies with rats, researchers found that high levels of maternal care, specifically tactile stimulation like grooming and licking, actually trigger a long-lasting epigenetic change. It increases the expression of glucocorticoid receptor genes in the baby rat's hippocampus. Basically, by getting more care early on, they physically grow more receptors. This means they grow up with better, stronger brakes for their stress response, making them fundamentally more biologically resilient to stressors later in adulthood. It's just incredible. Section 5. Calming the system, pharmacology. Let's move on and see how this builds into real-world applications. Because when the stress response becomes chronically hyperactive, how do we actually fix it? Well, our deep biological understanding directly informs pharmacological treatments. Since anxiety is fundamentally a hyperactive stress response, these drugs are designed to target the exact circuits we just learned about. Take benzodiazepines, for example, like Valium or Xanax. They work by enhancing the effect of GABA, which is the brain's major inhibitory neurotransmitter. It's essentially chemical suppression to forcefully calm those overactive fear circuits. Then we have SSRIs like Prozac or Zoloft. You probably know them as antidepressants, but long-term treatment with SSRIs actually increases the number of glucocorticoid receptors in the hippocampus. Just like that maternal care in rats we talked about, SSRIs help the brain build more brakes, strengthening that negative feedback loop to dampen the HPA axis over time. And finally, looking toward the future, we have CRH antagonists. These are experimental drugs designed to block the CRH receptors in the pituitary gland entirely, literally stopping the chain of command right at step 1. Which really leaves us with a truly provocative question. If we master the biology of the HPA axis, will we eventually use CRH antagonists to turn off the human stress response entirely? And maybe the better question is, should we? I mean, we've seen how damaging chronic stress can be, destroying the hippocampus and trapping us in this vicious cycle. But we also know that acute cortisol is a masterpiece of evolutionary survival, making us alert and capable in the face of genuine danger. As human bioengineering pushes further and further into the cellular mechanics of our own minds, we're going to have to decide not just how to calm the system, but whether we dare to rewrite the architecture of human panic itself. Definitely something for all of us to deeply consider. Thank you so much for joining me on this explainer, and keep questioning the biology behind the behavior.